Good day. Let's see, we are moving on. We have done various tests to test for the presence of different bacteria. We've looked at protozoa. Now we're going to look at the kingdom fungi, or the fungi kingdom, and we're going to look at yeast today, and we're going to look at the molds next week. Okay, so fungus, if you recall, is an organism that has a cell wall. It is an organism that's multicellular or single cell, depending on the species. They also decompose, meaning they break down organic matter, help recycle stuff. So when you see that mold growing on your fruit or something, they're decomposing it, is what they're doing. So what we're going to look at today is yeast. So in the fungus kingdom, there's two main groups of organisms. There's the molds and the yeast. The molds that we're going to look at next time are going to be multicellular, filamentous, things like that. Yeast are going to be the single-celled, and growing on a plate, they're going to look a lot like a bacteria colony. Okay? They're going to be larger because they're eukaryotic cells, but they're going to look very similar. Yeast divide very rapidly. Not as rapidly as bacteria, but pretty close. So when you put them in the incubator for several days, they don't like to grow at the same temperature as bacteria, so we're going to put them at a lower temperature, and it may take 4 days to get a really good colony, depending on the species. Like I said, things like Saccharomyces cerevisiae, which is baker's yeast, is going to grow pretty fast. So what we're going to look at today is how we're going to classify different types of yeast. So if you look at them on a plate, they're going to look like a bacteria colony. They're going to be round. They're all going to be usually mucousy looking, slimy looking colonies. You're not going to see a lot of distinctions between how they look on a plate. Some are going to have color to them, but most of them are going to be like that off-white color that we've seen with a lot of bacteria. One of the species that we're using, the Rhodorula rubra, it's going to be a pink color. It's kind of a pretty pink color. So for the yeast, what we're going to be looking at is some different characteristics. When you go to classify yeast, they're all single-celled. And basically we're going to classify them based on what they can ferment, so what type of sugar they ferment, just like we did with bacteria. And then we're also going to look at their different reproduction mechanisms. Most of them are going to have asexual, or most of them are going to have asexual reproduction. Some of them may also have sexual reproduction. When we looked at classifying molds, we looked at things like ascospores, where they produced it within a sac. So there is a sexual reproduction phase of some yeast, which is similar, where they actually have the sac, that the positive and negative cells fuse together. But also we're going to look at asexual reproduction. Yeast that are reproduced asexually are going to be either what are called budding yeast or fission yeast. The budding yeast, literally what it sounds like, a part will grow off of that yeast and then bud off into its own cell. That's asexual. And it's asymmetrical, meaning the parent may be this long and the little bud may be this big, and it's not a real pattern to it. The fission yeast are going to be more the symmetrical. They're going to kind of elongate and then split in half. Okay, but because they're eukaryotes, they're going to go through mitosis, just like our cells would do, but they split, almost like a bacteria would, in the sense of the cell wall, and into two equal cells. Okay, now because this is asexual reproduction, it's just mitosis. There's no meiosis involved in that. With the ascospores that are produced during some sexual reproducing ones, then that would be a type of the positive and negative cell coming together and making a new cell. Okay, so what we're going to look at is the 5 different species. We have Saccharomyces cerevisiae, which is your baker's yeast. We have Candida Albicans, which, if you've ever heard the word yeast infection, that's what causes that. It's a normal microbe that lives on the human body. The other one is that Rhodorula rubra, and another one we're going to use is the Selenatilla, all right, intestinalis, and the last one, Schizosaccharomyces octosporus. Okay, those are 2 new ones. I haven't seen them before in this particular class, so I kind of struggled with their names. Okay, so we're going to look at these in different ways. So the first thing that you're going to do is each group is going to have the 5 yeast. They're going to be growing on plates most likely. So they'll be on a specific plate that grows fungi. And then you're going to have fermentation tubes. So because you have 5 of them, you will have 5 fermentation tubes of each kind. Okay? And then you're going to have some sterile saline, and you'll have some other plates. So let's go ahead and just get started with the whole procedure, starting with the first part, which is just looking at the yeast under the microscope. So we haven't used this for a while, or have we? Not in a week, I think. Okay, so we got our flame going, and whoa, my thing's gonna fall over, which is not good. Okay, so the first thing that you're going to do is you're going to make a wet mount, just like you did with the protozoa, but this time you're going to do it with the yeast. Okay, so this time instead of putting water on the slide or anything for your wet mount, you're going to use this lactophenol cotton blue solution. And so it's going to be a light blue color, and you just put a small drop. It says a couple of drops, but 1 is fine. And then you're actually going to take a loop full of the yeast from the plate. Okay, and I'm actually not going to do this because these plates are sterile and I don't want to mess them up. So you're going to take the plate open. So usually we've gone from a tube, but this time we're going to come from a plate. So you're actually going to just open it up, and you're going to take a colony, and you're going to spread it into the blue solution that you have on there already. Okay? Flatten that, get rid of it. And then you're going to cover it with a cover slip. So you're not fixing the slide, you still want it to be a liquid. Okay? So you've just basically added the yeast to the blue solution so that you can give them color. So you're going to take that and you're going to observe all 5 of them. So you'd have a different slide for the 5 different species, and then you're going to look at the 5 different ones. Okay, so that's the first step. And then you're going to be looking for what do they look like, their size, their shape, do they appear to be budding or anything like that. The next thing you're going to do is the fermentation. I'm not going to demonstrate the whole thing again, you've done this before. Now I have here a red fermentation tube like we've used before, but in this particular lab, we're going to be using a different indicator which is called bromocresol purple. So instead of being red, it will be purple. And it still has the Durham tube in it to look for the presence of gas. So exactly what we did with the bacteria when we looked at fermentation, you'll inoculate the tube using all your sterile technique. You'll put it in the incubator a little bit loose so that it can get some air. But instead of being red, it will be purple, but you're going to be looking for the same results. It'll turn yellow in the presence of acid, and then gas will form inside that Durham tube if that yeast produces gas. Okay? Now for this, you'll have 5 of them. 4 different kinds. You'll have lactose, maltose, sucrose, 3. Okay? So you'll have a total of 15 tubes. You'll have 3 different sugars for the 5 bacteria. All right? Then the other thing that you're going to do, so that's the second part, and so you'll wait for fermentation for the next time. The next thing that you're going to do is you're going to have 2 plates of a agar called glucose acetate. It's a special agar that helps them grow and reproduce, and if they produce spores, you'll notice the difference. And you have 2 plates because you have 5 cultures. Basically, you take one plate and divide it into thirds, and you take the other plate and divide it into half. So you'll put 2 species on this plate and 3 species on this plate. In this particular one, what you're going to do, and I'm not going to show the whole procedure with the yeast, but you'll take your yeast and you'll put it into sterile saline, and so you'll use your aseptic technique just like you've done before. You put the yeast in here, and you're going to put quite a bit in there, and using your sterile technique, okay? And then you're going to kind of just tap it like we've done in the past, and you want to have a good cloudy suspension of yeast. And then you're going to take a drop of that. Let's open this guy first. So you're going to use a little pipette, and then you're going to take just a little bit of the yeast suspension that you've made, and you're going to put a drop of it. I guess I will mess up this plate. Oh well. So in the section where that one is labeled, you put one drop, just a drop. You don't spread it or anything of that particular yeast. Okay? And I'm just going to squirt the rest of it in there. So then you'd immediately put this into a sterile bag or the autoclave bag. And so you'll do that exact technique with the 3 on this plate and the 2 on this plate. Once again, you're just putting a drop on there. You're not spreading it because you don't want them to spread together, and they're going to spread out from that point. So if you'll notice, it says to allow for it to diffuse into the agar. So just let it sit. Put it off to the side. Don't flip it over. Remember how we've been putting things in the incubator upside down? We're not going to do that. We want that to just sit there, and then you're going to put them in the incubator. Now the incubator this time will be at a much lower temperature. It's going to be at 25 degrees instead of 37 because yeast like it a little bit cooler. Okay? So you're putting these in the incubator. You'll have 15 tubes. And then you'll have 2 plates, and you're going to put them in the incubator and come back and look at them next time. Fermentation, you'll be looking for changing color to yellow and possibly gas. For these, you're going to look for sporulation if it looks fuzzy, and then you're going to take a sample of it. So after these have grown for the week, and so if you're a 2-day class, you're not going to look at these plates for a full week. Okay, so if we start on Tuesday, you look at them the following Tuesday. And then from the plate, you're going to be taking a sample. So just like we did with the blue solution, this time we're going to use an iodine solution. The same thing, you're going to put a drop of it on the slide. You're going to add a loopful of the culture, put a cover slip on it, and observe. And this time you're really, really looking at the actual reproduction of the cells, because this particular agar will allow them to reproduce really, really well. So you should be able to see the differences between the 5 different species.